Hey, welcome everyone. Welcome to CHM Science. My name is Beth Foss, and I am the Director of Operations for the Foundation. Today, we're going to be meeting Kim Edwards, and we're not going to say doctor yet, but I know she's very close. She is on a great team at the University of Wisconsin, UW-Madison, alongside Dr. David Gamm. Kim is the first recipient of the Randy Wheelock Scholarship Award, and she's doing fabulous work that we're going to learn more about today. This is a series that we are excited to give to our viewers thanks to Genentech's sponsorship. And I know we have a large crowd with us here today, but welcome aboard. I bet it's a lot colder in Madison than it is here in Pennsylvania.
Absolutely.
We'll go ahead and get started. Thank you so much. And just to remind everyone, if you have questions, please use the chat feature. I'll probably keep most questions Kim and I talked about to the end, unless it's fitting to address them right at the time. But thanks to everybody joining us, and we really look forward to hearing more about your work, Kim. Thank you.
Thank you so much. Okay, I'm going to share my screen. You shouldn't see anything yet, but hopefully now you can see my slides.
We sure can. Yep.
Awesome. Okay. All right. So thank you for that really kind introduction. I am Kim, and I am so happy to be here and be able to talk about some of the work that I'm doing. And I'm really looking forward to telling you about kind of what I do, but then also what our lab does here at UW-Madison. So as mentioned, I am a PhD student in Dr. David Gamm's lab here at UW-Madison. And I'll be talking about how we can use, and what we're doing right now, to use stem cell technology for the study and treatment of choroideremia. Now, just before I start, I wanted to give a little bit of background about me and kind of why I came to this field and why I love it so much. So I actually graduated from Indiana University in 2014. And I knew I was really interested in going into a biology field, but I was really unsure of what research field I wanted to work in. And so I went ahead and I worked in industry for about 2 years, primarily using cancer cell lines and pharmaceutical drug development and testing. And this was really my first real hands-on experience using cells as a tool and as a model. And I just loved it. And so I decided to get exposed to some other avenues of cell-based sciences. And I took a course in stem cell technologies here at Madison Area Technical College, which is an exceptional course that got me some really great insight into stem cells and how to work with them. And from that, I actually made a connection with Dr. David Gamm here, and decided to join his lab to get some more experience in a research setting. And as you can tell, I just haven't left. So that was in 2017 when I joined, and I joined as a technician and transitioned my way into becoming an actual graduate student in the lab. So just to get us all on the, the same starting point, because I know this was something that, I had to learn along the way, was when people were talking about stem cells, what does that really mean? And what stem cells are is they are this really amazing, unique cell in the human body that has the potential to not only self-renew indefinitely, but actually also differentiate into more mature cell types. And when I say differentiate, what I really mean is that it has the ability to become different or specialize during the process of growth and development. And there are a couple different terms that go along with stem cells. So we have some stem cells that are unipotent, meaning that they have the capacity to differentiate into or become only one cell type. And there are also these stem cells that are multipotent, meaning that they can differentiate into cells belonging to more than one cell type. And to go a little bit further, there's this term that gets thrown around a lot, which I'm sure you're used to hearing, which is pluripotent stem cells. And these cells are actually able to differentiate into cells belonging to all 3 germ layers. So we're able to create cells that, from stem cells, they're able to generate ectoderm cells, which are cells, an example of that is your skin cells. endoderm cells, which example of that is your muscles, your bones, and then mesoderm cells, which are cells that typically line the inside of organs like your digestive tract. And so while there are different types of stem cells, and you can find unipotent and multipotent cells in different parts of your body, pluripotent stem cells are really found in the inner cell mass of the blastocyst in a developing embryo. And from these cells, we're able to generate an entire human being, which is pretty incredible. And in 1998, Dr. Jamie Thompson here at the University of Wisconsin actually was able to isolate these embryonic stem cells and grow them in a laboratory setting. And Dr. Thompson and his collaborators were then able to generate banks of these cells, so actually freeze these cells down for researchers across the world to use these for years to come. And our lab still does use these original embryonic stem cell lines that were created during this time for some of our research. And this was a huge advancement in stem cell biology before my time, but I'm very grateful for it. And as we're really increasing to understand, to be able to study human diseases in a dish, this was a really big advancement. In 2007, Dr. Thompson, in addition, Dr. Yamanaka, were actually able to kind of take this stem cell a step further. And they were able to generate what we call induced pluripotent stem cells, which are, you can take cells, human skin, blood, and actually reprogram them or return them to an embryonic-like pluripotent state. And these cells then have the capacity to become cells of all three germ layers, just like these embryonic cells. And so what's really fascinating and amazing about these is that we can now take cells from an individual, so say from me, or someone, with a disease or not, and something that we're interested in studying, and actually be able to generate specific cell types from these cells that are affected in this disease. And so our lab really takes advantage of a lot of this induced pluripotent stem cell technology, especially in the study of choroideremia. Now, I know that when I first got into the stem cell field, I didn't really know what to expect. And so I kind of wanted to go over, what does a stem cell laboratory look like? Because I think it's very cool, but maybe not as sci-fi as it sounds, or as at least as my family thought it was going to be. So a lot of individuals working in stem cell laboratories such as us spend a lot of time maintaining stem cells in our tissue culture rooms. So this is an example of one of the tissue culture rooms that we have here in the lab. And we actually spend a lot of time working in these biosafety cabinets, which is a fancy, word for like a space that we can work in that's sterile and keeps our cells from getting any bacteria or anything on them that would cause them not to be useful for us. And stem cells are really needy. And so anyone who works in stem cells know that we have to really baby them all the time to make sure they're happy and ready to be put to work in our research. And so another thing that we do is really keep our stem cells in something like this. This is an incubator, which actually is, we try and have an environment for these cells that really mimics the human body with a temperature approximating human body temperature and so on. And when we actually look at, stem cells, what we do is we actually grow them in these plastic plates that you can see here. And each of these has, this plate specifically has 6 different wells. And if we zoom in on a well, we can see that we have these almost island-like appearance to these cells. These are stem cell colonies here that actually adhere to the bottom of the plate and grow here so that we can then use them for actually creating other cell types. So when I work in David Gamm's lab, I'll just say that again, and he's a pediatric ophthalmologist by training, and he also is a professor in ophthalmology and visual sciences. And so when he was seeing a lot of patients come through his clinic and not have a lot of tools to be able to study a lot of the diseases that he was seeing, he really wanted to ask this main question, which was, can we utilize stem cell technology to really study inherited and acquired degenerative diseases of the retina? Now, the retina itself is made up of a variety of different cell types, and we generally focus on the outer retina. So you can see that, as light enters your eye, it hits this tissue back here, which is the retina. And the cells that we primarily focus on in our lab are these photoreceptors, which are the cells that sense light and convert it to a chemical signal so that that signal can then be sent to the brain. And these retinal pigment epithelial cells, which is this pigmented cell that actually provides a lot of support to the photoreceptors and keeps them happy. So in many of the retinal degenerative diseases, one or both of these cell types is affected. So it's really important that we're able to generate these cell types in a dish for studying these diseases. And another reason our work is really important is that not every disease that we're interested in studying has a great animal model for it. And choroideremia is a perfect example for that. I know that the CRF is working on generating a pig model for choroideremia, but there aren't too many options available for mammalian models for this disease, especially. So even though the photoreceptors and RPE are, grown together or sit together in the retina themselves, in our lab, we actually generate both of these cell types separately. And so I'm going to go through kind of how we make those cells before I talk a little bit about how we're able to use them for this, for studying choroideremia. So to generate photoreceptors, we take stem cells and we actually push them and create what we call retinal organoids, or these little spherical collections of retinal cell types. So these retinal organoids are actually what's housing our photoreceptor cells. And we push these stem cells to make these retinal cells using a cocktail of components that is found in the liquid media that we feed our cells. And as we're pushing them along this, we spend a lot of time taking care of them, and we actually are able to generate kind of these 3D like structures that actually are kind of the precursors or the beginning stages of what will eventually be photoreceptors. And we sit, this is my technician actually sitting, and she's dissecting and separating out manually these retinal precursors from remaining cell types in the differentiation process. And we take those, once they're little spheres, and we actually grow them and put them in flasks that are floating in a flask. And so this is kind of a representative picture of the flasks that we maintain, or how we keep our retinal organoids. This is approximately 50 organoids, so hopefully you can appreciate size of the little organoids, especially when you look at that compared to the size of a hand. And you'll notice that they're floating in this pink orange liquid. And I like to think of cells and cell culture kind of like fish in a fish tank. So fish get their nutrients from the water or the liquid, and then they excrete their waste back into that liquid. And so similarly, our cells obtain nutrients that from getting fed a liquid media that has a lot of components to keep them happy. And they excrete their waste directly in that liquid as well. So one of the main jobs of our technicians and other people in the laboratory is actually changing out this media in these flasks during the differentiation process every day to every couple of days, depending on where we're at. It's a big task. And like I mentioned, we spend a lot of time and care babying these. And actually for about 200 days, they're getting fed and maintained until they start to kind of look like this. So this is a retinal organoid. And on the outer rim, you can see these, it almost looks like a brush or like hair-like little structures that are on the outer rim. And what that actually is those are photoreceptors that are lining this outer portion of the retinal organoid. And so we're able to fix those organoids and actually take really thin slices of them and do some staining for some actual photoreceptor specific markers. So we can look at, do our organoids house photoreceptors? And we can compare that to something like a non-human primate thin section. And what we see is that these photoreceptors are really lining the outer rim of these organoids. And we have both rods and cones. So those are the two main photoreceptor cell types that are present in the human eye, ML cones and S cones and rods. And so this is a really great system for us to be able to generate photoreceptors in a dish for modeling. And as I mentioned that we have a separate method for creating our PE cells, so I'll go into that a little bit. And for our RPE cells are retinal pigment epithelial cells. We kind of go through a similar process where we take our stem cells and we push them towards generating these retinal epithelial cells. And over the course of about 60 days of differentiation, these cells that we plated down at the bottom of this dish start to take on, change their color, which I love. And I thought this was the coolest thing when I first joined the lab. So these cells in your eye are a pigmented cell type, meaning that they have melanin in them. So just like your skin has pigmentation in it, these cells do as well. And so as they're maturing, they really start to take on this dark brown black coloration that's characteristic of RPE, which is really cool. And we're able to then go ahead and separate out the retinal pigment epithelial cells and purify them and use them for modeling diseases and looking at function. So now I went through all the cell types that we typically grow in the lab and that we use for a lot of our studies. And I really want to talk about how these tools are being used to study choroideremia. So I couldn't talk about that without talking about the exceptional team that we have at in the Gamm lab studying choroideremia. So this is me, and this is my technician, really talented technician, Yoli, who has been with me for about a year. She does a lot of tissue culture maintenance, and of course, Dr. Divya Sinha, who has been an exceptional mentor to me and is actually working on another piece of the puzzle for choroideremia with me that I won't get to talk about today, but that, she's been instrumental in this. And I kind of took over this project from her when I became a graduate student. So she is still involved heavily and helps a lot. And then her technician, Cole, who has been helping a lot with this project as well. So in choroideremia, when we look at how the disease progresses and, what happens in these cells over time, so we know that the typical or the supposed site of degeneration is really the photoreceptors and the retinal pigment epithelial cells. Now we have this underlying choroid, which seems to be kind of a secondary degeneration in this disease. But as the disease progresses, we're losing these cell types as they become unhappy and start to die off until this late stage where we really have very few cells left in certain parts of the retina. Now, it's really difficult to use one approach to target all stages of disease progression. So in early stages, when you still have the cell types there, we're all working on prevention methods and different pharmacological therapies and gene therapy as well. But in these late stages, when the cells aren't there anymore to be able to, administer these therapies, cell replacement therapy becomes a really important approach. Now, I know that I mentioned gene therapy here. And with the disappointment from the recent gene therapy trials, researchers like me and my team are really trying to get back to studying the basics of this disease to try and shed some light on what is really going on during the disease progression. And we're hoping that this will give us some idea on how to guide some better strategies for therapeutics, especially in these early stages of disease progression. And so focusing on this part, my goal for a good portion of my actual studies here in the lab is to better understand the basic biological mechanisms of disease. So we couldn't talk about choroideremia without talking about the protein that's actually implicated in the disease. So Rab Escort protein 1, or REP1, which is how I'll call it for the rest of the time, is a protein that is encoded by the CHM gene. And it's a protein that's found in every cell of the body. And what's interesting about this is that, as we all know, it's really interesting that the main site of disease pathology is in the eye, even though this protein is found in every cell of the body. Now we know that there are over 140 mutations in the CHM gene, and all of these are predicted to result in a loss of function of REP1. But to really know what, a loss of function is going to do, we have to know what the protein is doing in a functioning cell. So we know that REP1 is required for proper functioning of another protein called Rab. And I like to think of this protein, REP1, as kind of like a taxi, and Rabs like individuals trying to get to work. So REP1 is really required for Rabs to be able to do their job. So Rep1 picks up these Rabs proteins and helps them get to where they want to go. And what they want to do is really regulate transport within the cell. And I know that this is a very busy graphic. I actually really like it kind of for that reason. So what I want you to take away from this is how complex this is. But the main thing is that we know that there are over 60 different Rabs that have been identified in a mammalian cell so far. But what we don't know is which Rabs Rep1 actually interacts with in the eye, and potentially why does the lack of Rep1 in choroideremia cause unique disease pathology of the eye due to these interacting partners of Rep1. And so a large portion of a lot of my work right now is to try and figure out what the interacting partners are of Rep1. So identify, what the Rabs are in the cells specific to the retina, so RPE and photoreceptors, and comparing that to non-retinal cell types and seeing if there is a difference that can shed some light into how the disease is progressing. And as we're working on that part of the puzzle, we're really working on testing therapies for these early stages, and hopefully this will kind of guide some of our next steps. Now I want to talk a little bit about what we're doing for some of the later stages of this disease. And this is really work that was started and is ongoing with a fantastic team that I'll call the transplantation team. So this is our cell replacement team, or the people that are really working on some of these later stage disease options. So what this team does is we take our ability to generate these human stem cell derived retinal cells, and we actually take them and we transplant them into other species. So what we're hoping is that this is, kind of a preclinical model for being able to actually transplant cells back into patients that have lost these cell types. And one challenge that we're finding, which is where I come in, is that the species are different. Who knew? And these cells from different species might not be able to talk to each other. And so even though we're putting these human cells in, they might not be able to make connections with the host cells that are actually there. So while we can work on our transplantation strategies, look at safety and optimize a lot of our techniques in these other animals, we really might not be able to get exactly what we want, which is that the human cells we're transplanting will actually make connections with the cells that are already there and restore vision. And so to overcome this problem, we have a really large grant through the Department of Defense in collaboration with a lot of different researchers across the world, actually, where we are taking out the potential species differences, issues of transplanting human cells into pig, and actually generating retinal cells directly from pig for transplantation. And so that is another piece of some of the work that I do in the lab. And primarily, my goal is to generate photoreceptor cells from these pigs. And so to be able to do this, we needed a really great pig-induced pluripotent stem cell line. And so luckily, we have a fantastic collaboration with Dr. James Thompson and one of the scientists that was previously in his lab, Dr. Li-Feng Chu, who now has gone on to start his own lab in Calgary. But they were kind enough to send us their newly derived pig line, or newly generated pig line. And I worked through and tried to make a method to actually be able to kind of repeat what we do in humans with these pig stem cells. And so I was successful in being able to create, these little spheres, these little retinal organoids that are very characteristic of one part of our human protocol. And when we take a closer look at those, so fix them and take those really thin sections again and look for photoreceptor, are we generating photoreceptors that we could potentially use for transplantation? We see that these spheres are just chock full of photoreceptors at the outer rim, just like we're seeing with our human organoids. And so this was really encouraging. And so a lot of work on this part of the project is trying to better characterize these cells. And then hopefully in the next few months, we'll be able to transplant those into pigs and see what that'll do. So that's really exciting. And I'm really happy to be part of this part of the project as well. So I know I threw a lot at you, but hopefully I got across that stem cells are a really powerful tool for generating human retinal cell types, and especially for diseases like choroideremia, where there are really limited mammalian animal models. And that our approach really is to study, across the disease spectrum and try and generate therapies or better understand what's going on in different stages of the disease. So really getting back to the basics for these early to mid stage of the disease, and then focusing on developing these cell transplantation efforts for late stage of this disease. So with that, first off, I want to say thank you so much to the Choroideremia Research Foundation for all of your support. I am so grateful to not only be the first recipient of the Randy Wheelock Research Award, but just be able to participate in all of the different meetings and meet with all the different researchers and all of you. It's really helped me have a really great understanding of this disease and constantly be learning about different aspects as I progress through my studies. I also, of course, want to say thank you to Dr. David Gamm, who is the best mentor. I couldn't ask for another one, and hence why I'm still here. And then, of course, Dr. Divya Sinha as well for all her mentorship on this project, and the two technicians that work really hard, Cole and Yoli. We have a huge lab here at UW-Madison. I don't even think this is all of our members. Because of COVID, we haven't been able to take too many group shots anymore, but we're about 20 strong at this point. So thank you to all of them for all their support, and just being the greatest lab environment that I love going to every single day. So with that, I'll take any questions. Thank you so much.
That was awesome. That was really informative. So now's the time. Oh, here they come. Here come the questions. Somebody asked earlier, you'll be joining us right in June.
Yes, I will.
That's great.
I'm really looking forward to it.
It'll be a wonderful time. And that's a good time right now to plug the conference, everyone. June 15th through 18th, go to www.curechm.org. And it's really easy. There's a spot right there on the website that you can get more information and link it right up to the app, which will take you to the registration. Okay, here we go. This comes from Brent. Hey, Brent. Wow, incredible and thorough presentation of the Stem Cell Avenue. Great work, Kim. Yay.
Thank you.
Can Kim estimate when the team will conclude either success or not success on restoration or improvement of retina performance in a pig model?
That's a great question. So it's definitely kind of a long process. So right now I'm still in the stages of doing a lot of the work in-house. And so hopefully we'll be transplanting some of the photoreceptors that we derived from the pig stem cells into pigs in the next few months. At that point, it'll probably take, I would guess, a year or two to actually really fully figure out how things are integrating into the retina. Are we getting restoration of function and everything? And since there's so many different teams that are involved in this, so the National Eye Institute actually houses all the pigs. So we have a big collaboration with them. And so it'll probably be another year or so, I think. We'll have some good answers in the next few months of just how it's going, but it's going to be kind of a process. But hopefully it'll still give us some really good information on our ongoing human work, and just making sure that we can hopefully get some connections between these cells. And I didn't go into it in big detail, but hopefully, yeah.
I think that was another question one had, and you touched upon it now, or just the next steps, kind of the collaboration. Do you or Dr. Gamm have expectations as to whom you will be working with in the future in those next steps?
Oh, so especially for the pig work. So we have a lot of people that are involved in that big Department of Defense grant. And so there are people at the National Eye Institute who have the pig models. Also, they are trying to generate the retinal pigment epithelial cells, or RPE, from those pig cells, because that's kind of one of their expertises is RPE in the lab that we're collaborating with. And then also we have collaborations with members of the military in other countries that are helping us actually with the Department of Defense. I didn't go into that too much, but the Department of Defense is really interested just because a lot of individuals who are in the field and in the military actually get injuries based off of blasts or having lasers shown in their eyes. And so they tend to have this really unfortunate loss of vision when they come back. And so that's why they're really interested. And so we're hoping to kind of use that as a tool for advancing our studies here. So, and Dave also, I should have mentioned, Dave has a company that he does a lot of work on. It's called Therapeutics. And so he's trying to advance that photoreceptor cell replacement therapy with that as well.
Awesome. Here comes another from Mary. Kim, can you give us a high-level description of how induced pluripotent stem cells or iPSC cells would be transplanted into the eye?
Sure, I can give you a very high level because I do not do that transplantation myself. So some of our transplant team would be better people to answer that. But actually, we don't transplant human-induced pluripotent stem cells or stem cells directly into the eye. We make sure that we differentiate them towards retinal cell types first. And our lab actually has a lot of different methods of doing it. But one of the main ones that we recently published a paper on was actually creating these little tiny biodegradable scaffolds that we can actually take our cells and put them on that scaffold. And then it's kind of like a little sheet that we can put in the eye. And so it's a subretinal transplant. And so we kind of separate out the retina, transplant that in, and then, yeah, so that's how we actually put it in. But I can't give you too many more details just because I'm not the one doing it, but.
Good that you guys are doing that, right? That sounds very technical. Wow.
Yeah, and that's why they're working with a lot of different animal species to test kind of the beginning stages of that. So hopefully we can have a good idea of how to proceed with that, and then hopefully it'll move on to some clinical trials.
That's very encouraging. Here's another one from Kevin. Will this research path likely lead to stopping the loss or progression, or might it lead to restoration of retina performance for those who have already suffered significant loss? Thank you, Kevin.
Sure, great question. So kind of the research plan that we have for the beginning stages of the disease, what we're really hoping is that we can come up with some, by just knowing a little bit more about how this disease progresses and what the protein that's involved really does. Hopefully we can come up with some therapies that will actually halt progression of disease or kind of slow it down or prevent it from advancing too far. For people who are already kind of further along in the disease, that's where that cell replacement therapy comes in. And so in that case, we're hoping that we can restore some vision for those individuals.
A lot of our efforts as an organization has been put behind stem cell therapy and gene therapy. And so this is encouraging to give some of the people who have more advanced cases of CHM some encouragement and hope, and site replacement's really big. And we'll be having lots of speakers on that in June. Let's see, is there any other questions anyone has? You know, and we'll give people a few more minutes, but we always like to ask this. Is there anything outside the lab or outside science?
I think you cut out a little bit, Beth. Could you repeat that, Beth? I'm sorry, you cut out a little bit for me.
You know what? I just got a thing. I don't know. Our internet's unstable. We have high winds here on the East Coast today. I apologize.
No problem.
Is there anything fun outside the lab or science that in your personal life that you like to do that's fun? We always ask that of our scientists.
Yeah, that's a great question. So now that I'm a graduate student, I don't have as much time to be outside of the lab, but I actually do improv comedy, which is really fun. And I also do tap dancing and a lot of yoga. So yeah, so those are the things I really enjoy doing outside of the lab and as a nice relaxation.
That might be fun. Maybe you can do a little bit of that in June with us.
Yeah, that would definitely be exciting. All right.
I know
Only people
Corey,
with me.
Corey would ask that question. I know he's already thinking and planning. So Corey, don't worry, we got it covered. He's
Good,
joining
good.
us. Oh, Brent Pike is always one. He's on with us almost every month, and he always adds a quick little funny thing. He said, "Are Canadian pigs slightly more polite to work with?"
I don't know, but I feel like they must be, right? I don't
Right?
personally work with pigs, but I feel like they have to be. So
I don't know. I think Wisconsin and Minnesota people tend to be pretty nice up there, eh?
yeah, exactly. Yeah, it's true.
Oh, yes, they want a demonstration of your tap dancing skills, so please bring them.
Okay, good. Yeah. I'm not saying I'm an expert, but
Well, it'll be fun. It'll be wonderful. Hey, Dorothy's asking another question, back on task. Is stem cell transplants currently taking place in humans? To what degree of success comes with it?
sure. For especially, I'm going to focus on retinal transplants. So right now, they're especially for the photoreceptor side of things, they're not currently taking place. There have been some studies on especially the retinal pigment epithelial side of things, kind of trying to get that to be more of a mainstay. But that's definitely something that Dave and his company is working on, is trying to do a lot of the preclinical work, so a lot of work in animals to try and kind of validate those studies and then transition them to humans in the next few years. So.
Well, I think that's it. Just want to make sure if anyone else has any last questions. This was really good. It's very encouraging, and we always like hearing more about the money that people are raising and fundraising to bring to the research. It's really important that people know what it's being used for. We have a very robust science advisory board, which meets monthly, and they review the different science and the prospects. So it's all it's moving forward. We're really excited. Oh, here's another question, Dorothy. When do you expect a clinical trial to take place? We always try to do that. We know we want to try to, you don't have a crystal ball, we understand, but it's always, we'd like to manage our expectations of our members. That's something that's pretty tough to do, but do you see that? Kind of answer that in part already, but do you want to take a stab at it?
Yeah, I unfortunately don't think I can really answer that question, like give you a exact time or when. And I know that's frustrating, but I think something that I really appreciate about when I talk to Dr. Gamm about, clinical trials and pushing this forward is really that we're doing everything on our end to make sure that it's as safe as possible and making sure that we cover everything on our end. So I know it's not as fast as everybody would want. I mean, we're, in the same boat. We really want it to be pushed forward as well. But yeah, I don't think I can give you an answer to that. And I know Dave is going to be at the conference in June. So that's a good question to ask him because he's more in tune with a lot of that work. So.
Right. They always get asked that question. So we'll be sure to do that for sure.
Yeah.
All right. Well, I think that's it. I think that'll wrap it up, everyone. Thank you for joining. Thank you, Kim. Thank you so much.
Thank you so much. This was lovely. So I look forward to meeting everybody.
How much snow do you have left up there?
How much snow? It's pretty melted at this point, but I think we're getting some snow today. So we'll see. But we have a nice layer of ice that'll be underneath that snow. So it's going to be yeah, hopefully we'll be transitioning to spring pretty soon.
Soon, right?
Yeah.
Well, thank you, everybody. Thanks for joining us. We hope you have a great weekend. We'll be having another webinar next month, we're hoping. And so we'll look forward to seeing you then. And thanks so much. We want to make sure we say one more time, thank you to Genentech, who is sponsoring these webinars. And we look forward to seeing you next time. Thank you.
Thank you.
Bye, Kim.
Bye.